Thank you all for attending the webinar today. My name is Brady Strittmatter, and I'm Scientific Product Manager at VectorBuilder. VectorBuilder is a global leader in gene delivery technologies, and today I'm excited to present to you one of those gene delivery technologies, miniVec, VectorBuilder's novel miniaturized plasmid that is enhancing gene delivery. Traditional plasmid backbones are typically a few kilobases in length and include an origin of replication and a marker that allows for the plasmid selection in their host bacterial strain. While traditional plasmids have been a staple of molecular biology and the workhorse of many experiments and therapies that require gene delivery, they do have drawbacks that can hinder their effectiveness and safety. One of the main drawbacks of traditional plasmids is that they require antibiotic resistance genes for their selection. While effective, antibiotic resistance is a major public health concern, and antibiotic overuse is a contributor to the problem. In addition, there are regulatory guidelines for antibiotic use when manufacturing cell and gene therapies, which can limit their use in clinical settings. And the introduction of traditional plasmids in vivo also introduces the risk of gene transfer, in which bacteria in the host strain obtain the antibiotic resistance gene, which can contribute to the problem of antibiotic resistance. In addition to issues with antibiotics, traditional plasmids have size limitations partially imposed by the size of their backbones, which can contain a few kilobases of bacterial-derived sequences. Certain plasmids may also be difficult to obtain in high yields, which can lead to expensive and costly, time-consuming downstream purifications, resulting in less than pure products, which results in variable expression in experimental conditions. Finally, too high of a concentration of nucleic acids can be cytotoxic to cells, which can lead to problems with gene expression. In order to address the problems associated with traditional plasmids, VectorBuilder scientists developed miniVec, novel antibiotic-free miniaturized plasmid. The miniVec backbone can be as short as 500 base pairs and is derived from sequence with minimal bacterial origin. The backbone relies on a selection mechanism that is completely antibiotic-free, and the production of the plasmid does not require fermentation with any additional media additives or supplements. Figure 1 demonstrates the mechanism of selection with the miniVec backbone. The backbone consists of an origin of replication and a selection sequence, VecSeq-B. When VecSeq-B is transcribed, it can bind to a complementary sequence, VecSeq-A, which is upstream of a growth-inhibiting gene. Any of the mini-host bacteria that are not transformed with this plasmid will not survive, any bacterial host, mini-host transformed with this plasmid will survive and proliferate, and any bacteria not transformed with the plasmid will not be able to grow. I will further elaborate on the mechanism of the antibiotic-free selection with miniVec in coming slides. In addition to these features, I will also show data in this presentation that demonstrates that the miniVec backbone results in increased plasmid yields and transgene expression when compared to traditional plasmids. Due to miniVec's increased safety, efficacy, and manufacturability, we believe that miniVec is well suited as a backbone for cell and gene therapy applications, and it can be used across a wide variety of expression systems. I hope the data that I show you today will help convince you that miniVec is an ideal choice for these applications. Figure 2 is a comparison of yields from lab-scale fermentation of the indicated expression systems with either a traditional or miniVec backbone. As we can see from the results, the miniVec plasmid backbone exhibits increased yields across all expression systems compared to the traditional plasmid. There are multiple mechanisms by which the miniVec backbone could increase plasmid yields, including a reduced metabolic burden on the host cells, more efficient replication, improve cellular uptake and trafficking, or most importantly, the miniVec system has been engineered to favor a high copy number fermentation. Figure 3 shows a more detailed mechanism of how the miniVec system works. miniVec operates within the exclusive host bacterial strain minihost, whose genome has undergone strategic engineering to include a growth inhibiting gene, which is inhibited by a specific media supplement. This growth inhibiting gene has also been modified to incorporate a sequence called VecSeq-A upstream, positioned at a 5' end of the growth inhibiting gene. During transformation, the supplement is removed, and those mini host cells that harbor the miniVec plasmid will transcribe VecSeq-B, which VecSeq-B will then complement and bind to VecSeq-A, which will inhibit the growth inhibitor and allow the mini host cells to survive and proliferate. Any mini host cells that are not cultured with the supplement or are not transformed with the plasmid will not survive and proliferate. This mechanism can also favors high copy number fermentation, because the more copies that the miniVec plasmid that the host strain has, the better it will inhibit this growth inhibiting gene, and thus will survive and proliferate. Due to this design, miniVec can increase the manufacturability of cell and gene therapies. In addition to increased plasmid yields, miniVec also exhibits increased transgene expression in vitro. For figure 4, in this experiment, the same GFP transgene construct with either a traditional or miniVec backbone was transfected at an equal molar concentration along with an mCherry expression plasmid as a transfection control. From the data, we can see that 48 hours after transfection with miniVec, miniVec outperforms the traditional plasmid in transgene expression. GFP expression for miniVec is significantly increased for its relative fluorescence intensity. Potential mechanisms to explain this increased expression compared to traditional plasmids include enhanced cellular uptake of the plasmids due to their size. In addition, miniVec plasmids could reduce host cell stress by having less bacterial sequence to minimize the cellular innate immune response to foreign sequences. So miniVec's backbone allows for a more streamlined design with fewer non-essential elements and can result in increased transgene expression in vitro. Further, in addition to miniVec can also demonstrate increased transgene expression in vivo. In this experiment, the same transgene, luciferase transgene cassette with either a traditional or miniVec backbone was injected intravenously into mice at an equal molar concentration. The transgene expression was monitored by luciferase activity, and we can see a significant increase in luciferase activity with the miniVec backbone compared to traditional plasmids. Extending this validation data further, we can see that with the same type of experimental setup, but with an intramuscular injection instead of an intravenous injection, the miniVec backbone exhibits increased transgene expression compared to a traditional plasmid. In addition, we can also see that 3 of our 4 mice exhibit robust transgene expression up to 21 days post-injection. This in vivo expression for intramuscular injected plasmid, this plasmid was only co-injected with PBS. And this prolonged expression from this miniVec plasmid without the need for a transfection reagent allows miniVec to be a prime candidate for DNA vaccine applications. DNA vaccines offer several significant benefits compared to traditional vaccine approaches. Firstly, they are inherently safer as they do not involve the use of live pathogens, reducing the risk of causing diseases or any adverse reactions. Additionally, DNA vaccines are highly stable at room temperature, simplifying storage and distribution requirements. The versatility of DNA vaccines allows for their rapid development and customization, with a wide range of infectious diseases possible to target by just modifying the DNA sequence. And even DNA vaccines are being investigated for cancer treatments and cancer vaccines as well. Furthermore, DNA vaccines have the potential to induce humoral and cellular immune responses, providing broader and longer-lasting immunity compared to some traditional vaccines. Overall, the safety, stability, and versatility and the immunogenicity that DNA vaccines provide position them as a powerful tool to fight against infectious diseases and cancer and for the development of novel therapeutic strategies. And we believe that miniVec is a prime backbone candidate for these DNA vaccine applications. In addition to regular plasmid expression, miniVec can also be applied across multiple expression systems, such as transposon systems. In this experiment, both the piggyback transposase and the transposon were put in either traditional or miniVec backbones. An equal molar amount of each plasmid was transfected into cells, and at passage 1, we can see that the miniVec backbone results in enhanced transposon expression, but at this point, we can't really measure that as take that as a measure of transposition efficiency. So therefore, we passage the cells to passage 9, and when we see at passage 9, we see increased transposon expression and transposon efficiency compared to traditional plasmids, meaning that miniVec is a viable backbone for piggyback applications. And further, if we extend and do the same experiment using Sleeping Beauty transposon system, either with the transposase and transposon in the miniVec backbone or in a traditional backbone, we can see similar results with the Sleeping Beauty system. We see increased transposon expression at passage 1, which is not really a measure of transposon efficiency. However, by passage 9, we can see that the miniVec has outperformed the traditional plasmid backbone, meaning that miniVec allows for increased transposon efficiency and transposon expression in both piggyback and Sleeping Beauty systems. An interesting application of this that we've seen at VectorBuilder is for generating lab-grown meat strains, and this application is suited for commercialization options like that, as well as regular research settings as well. In addition to applications in transposition systems, miniVec can also be effectively utilized in recombinant viral systems, including lentivirus and adeno-associated virus, or AAV. The miniVec backbone has been shown to enhance the efficacy of these viral systems, specifically when packaging and transfer plasmids are all with the miniVec backbone compared to traditional backbones. We can see that a comparison of the packaging plasmids with the miniVec backbone and the transfer plasmid with the miniVec backbone compared to traditional backbones you get increased viral titers when using the miniVec backbone. As a result, researchers can achieve higher yields of recombinant virus, which is crucial for various applications, including gene therapy, functional genomic studies, and the development of viral vectors for protein replacement. This makes miniVec a powerful tool in the realm of viral vector production and offers a robust alternative to conventional traditional plasmid backbone systems. In addition to increased viral titers, lentivirus packaged with the miniVec backbone also exhibits increased transgene expression following transduction. As a result, lentiviral vectors produced with miniVec can deliver therapeutic genes more effectively, leading to higher levels of gene expression in the target cells. This advancement could be particularly beneficial for gene therapy applications, where robust and sustained transgene expression is crucial for therapeutic efficacy. miniVec applications of lentivirus could be extremely useful in ex vivo cell therapies, where high expression of transgene levels need to be sustained, and miniVec could act as a safer source material for this virus packaging with its antibiotic-free backbone. Ex vivo cell therapy involves genetically modifying patients' own cells outside of the body, commonly using lentiviral vectors to engineer these patient cells. This diagram here diagrams one application of this ex vivo cell lentivirus technology, which would be in the application of Zynteglo, a lentivirus-based ex vivo cell therapy for the treatment of beta thalassemia. In this genetic disorder, patients' stem cells are harvested, followed by transfection with a viral vector containing a functional copy of the beta globin gene. And this gene is introduced into patients' hematopoietic stem cells and allows them to restore production of functional, healthy red blood cells. This therapy offers curative treatment to beta thalassemia and addresses the underlying genetic causes, providing long-term clinical benefits, including a reduced dependency on blood transfusions. So with miniVec's enhanced genetic architecture and antibiotic-free backbone, we feel that it's a safer source material for protein replacement and gene therapies using viral vectors, both for protein replacement and for even potentially CAR T applications. So just to reiterate, for therapeutic applications, miniVec provides enhanced safety with its antibiotic-free backbone. It can help improve the manufacturability of cell and gene therapies by facilitating higher yields, which facilitates a better purification efficiency, resulting in cost-saving and time-saving advantages in the large-scale production of gene therapies. And then also, hopefully, some of the data I've shown you so far has demonstrated that miniVec can also provide enhanced efficacy for cell and gene therapies, as it can produce increased transgene expression both in vivo and in vitro. Just a little background about VectorBuilder and how you can partner with VectorBuilder to scale up your cell or gene therapy or for your research grade application. About this, VectorBuilder is a comprehensive CRO and CDMO, and they provide a full suite of services for therapeutic and process development in the fields of cell and gene therapy. VectorBuilder offers end-to-end solutions from vector design and construction to preclinical development and GMP manufacturing. So whether you wanted to investigate miniVec in a research grade setting, a preclinical setting for constructs that have already been investigated in the lab or are looking for a partner for commercialization of novel backbones or therapeutics. VectorBuilder can offer the full spectrum of solutions for gene delivery. These GMP manufacturing capabilities extend to cell and gene therapies, which also include miniVec. And our team is experts in process development and scaling up cost-effective production of high-quality plasmids and viral vectors suitable for clinical applications, as well as research-grade applications as well. VectorBuilder can also provide GMP manufacturing of these miniVec plasmids, which could help with the regulatory process as well as they are antibiotic-free. All of this technology is backed by VectorBuilder's Vector Design Studio. And VectorBuilder provides an innovative web platform that merges advanced vector design with seamless online ordering. The Vector Design Studio allows users to easily customize vectors and vector components like promoters, open reading frames, and markers. And the platform enables unlimited customization, such as sequence editing, tag addition, codon optimization. And additionally, users can order custom cloning of their vectors and downstream services like virus packaging and plasmid DNA preparation directly from the platform. So in addition to the miniVec backbone, which I'd highly encourage everyone to try out, VectorBuilder also offers a wide variety of plasmid backbones, comprehensive solutions for anyone looking to deliver a gene. Following completion of your custom vector design, the Vector Design Studio will spit out a vector information page, and you can share this design with your colleagues, order downstream preparations and services for it, such as plasmid DNA preparation or virus packaging. And users can also explore other vectors that VectorBuilder users have designed, allowing for both custom cloning ordering and a wide database of pre-made vectors which are ready for experimental use. So hopefully the data that I've shown to you in this talk will help convince you that miniVec is a prime candidate suited for cell and gene therapy applications, and that VectorBuilder is willing to partner with researchers to allow them to use the miniVec backbone to increase the safety, efficacy, and manufacturability of their cell and gene therapy products. I wanted to thank everyone for their time and participation in the seminar today. So thank you very much. Kamsahamnida.